In this lecture, I'm going to describe the histological structures in the ear. At the end of the lecture, I would like you to understand that there are functional divisions and anatomical divisions of the ear, that there are membranous and bony labyrinths that play a very important role in the function of the ear. We're going to look at various receptors of the ear, in the inner ear. They're hair cells, and they're very similar, except for those in the organ of Corti, a component of the cochlear duct, which I'm going to describe as well, and that you should have a thorough understanding of, because this is the sensory component that allows us to hear. We're also going to look at other parts of the ear that help us to know of our position in space and our head movements. We have in our ear a number of different components, anatomical components. We have the external ear and the middle ear that are designed to receive sound energy, sound waves, and then to amplify those and create or turn that energy or those sound waves into mechanical energy. And then we have receptors in the various locations in the inner ear that I'll describe that then use that mechanical energy in the form of vibrations and movement of fluid to then detect not only the position of our head in space and movement of our head, but also that mechanical energy is converted by the organ of Corti into energy that allows us to then hear. And that's all processed by the central nervous system. So we're going to look at all those very special receptors that help us hear and also maintain balance, equilibrium, and now our position in space. Let's first of all look at the general anatomical structure of the ear. On this diagram, there are various labels that do illustrate all the major components of the ear. Starting on the left-hand side is the pinna of the ear. That part of the ear is very flexible. We can twist and bend it and it will spring back to its normal shape because it's got elastic cartilage in it. That helps to locate or amplify the sound, but has very less function in humans than it does in other animals. The external auditory meatus is about 2.5 centimetres long. It is bound on the most lateral part by the pinna of the ear. And the very first third is supported by cartilage, and that cartilage blends with the elastic cartilage in the pinna of the ear. The skin lining the external auditory meatus has got special glands in it called ceruminous glands. Those ceruminous glands secrete a component that combines with apocrine secretions to produce earwax. And that earwax is used to try and trap any particulate matter that happens to find its way into that auditory canal. And that earwax is also suspended on the fine hairs in that canal, again to increase the opportunity to trap that matter. On the medial aspect, or the end part of that auditory tube, is the tympanic membrane, or the eardrum. It vibrates in response to sound waves. And those vibrations are passed on to the bony ossicles, the malleus, incus, and stapes, illustrated in the diagram. And those bony ossicles will vibrate themselves, and those vibrations will then be passed to the oval window in the inner ear. The oval window and the round window play a very important role in the functioning of the inner ear. You also have labelled there the auditory tube. It's a passage going from the middle ear cavity all the way down to the pharynx. Normally it's closed, except when we swallow or maybe when we yawn. It's a passageway or a potential passageway for pathogens passing up from the pharynx. Here are two diagrams representing the structure of the inner ear. On the left-hand side, you have what we name the bony labyrinth. It's the bony portion of the ear embedded in the temporal bone. Inside that bony labyrinth is going to be perilymph fluid. And I'll come back to that diagram in a moment. On the right-hand side, you have the membranous labyrinth, and that membranous labyrinth is filled with endolymph. Now what I want you to do is place that membranous labyrinth inside the bony labyrinth. And that's exactly what the structure, the anatomical structure of the inner ear looks like. Two components, the bony labyrinth filled with perilymph, and embedded in there you have the membranous labyrinth filled with endolymph. Let's turn back to the bony labyrinth and let me point out a few anatomical components. First of all, you have the vestibule in the centre. You can see the oval window and the round window. That vestibule contains the recess for the utricle and the sacral. The utricle and the sacral is one of the anatomical components of the inner ear that houses the receptors for movement of the head, linear movement of the head and the position of the head. They house the macula, and I'll talk about those later on in this lecture. On the left-hand side, you see the semicircular canals. They are at right angles to each other. There are three of them there. Those semicircular canals, as they approach the vestibule, have a dilated portion we call the ampulla. It's in the ampulla that we have special structures called cristae. And those cristae house the receptors for turning the head, moving the head at an angle. And I'll talk about those also in this lecture. Now, the third anatomical component of the inner ear is the cochlea, shown here. The cochlea is a spiral-shaped organ. It spirals about 2 3/4 turns. And down the centre core of the spiral is a bone called the modiolus bone. And right in that bone is the spiral ganglia. The spiral ganglion houses the nerve cell bodies, the neurons, the bipolar neurons, that are going to send fibres to the hair cells in the cochlea and transmit information back into the central nervous system that give us the sensation of sound. This is another diagram to explain what I mentioned in the previous slide. In fact, this shows you the membranous labyrinth and just a little bit more detail that I'll briefly summarise here. Focus on label 1 there. That's the vestibule. It's the vestibule of the membranous labyrinth, which, remember, is enclosed or encloses endolymph. You can see the macula saculi and the macula utriculi. And those two maculae contain sensory receptors for the position of the head and linear movement. 2 there, labelled, shows you the semicircular canals, and a little illustration at the top just reminding you that this membranous labyrinth is enclosed in the osseous labyrinth and surrounded by perilymph. As I explained, each of those semicircular canals has a dilation called the ampulla. And within each of those ampulla are the crista. crista ampullaris. They're receptors that respond to the angular movement or tilting of the head. So there are three of these and two maculae, and then finally the organ of Corti, the cochlear duct, create the 6 sensory regions of the inner ear. And the organ of Corti is going to be in the cochlear duct within the cochlea, and that's going to be responsible for sending sensations back to the central nervous system about sound. Before we move on and look at those sensory structures in more detail, it's important, firstly, to make sure we understand the histology and the ultrastructure of the hair cells. There are 2 hair cells shown here, type 1 and type 2. Hair cells are very similar, whether they're in the crista ampullaris or in the maculae. They differ in the organ of Corti in one way that I'll mention at the end of this slide. But essentially they're very similar. Type 1 and type 2 hair cells, I'm not going to really talk about any functional difference between these cells, but you can look at the slide and know that they're structurally different. The type 1 hair cells have a more flask or bulbous type of appearance or shape. The type 2 hair cells have a more elongated shape. At their apex, they both have projections called stereocilia. And notice that these stereocilia are different in length. They have a graded increase in length from one side of the cell to the other, and that's repeated along all the hair cells. And on the left lateral side of each of these hair cells, you see one cilium. It's called the kinocilium. That's a very important structure because that defines the polarity of the cell. What we're going to learn later on is that those stereocilia are actually affected by movement of the endolymph when we move our head in certain directions. And that movement of the endolymph actually moves those stereocilia towards the cilium or away from the cilium. And that sparks off opposite impulses. And it provides information about the movement of that fluid and therefore the position of our head. Those hair cells are supported by supporting or sustentacular cells. You can see they surround the hair cells. They're coloured a very light grey in this picture. And then the hair cells are innervated. The bipolar neurons from the vestibular ganglion send out a dendritic branch, the afferent nerve fibre you see here, that wrap around or form a bulbous type of connection to the type 1 hair cells, or simply just have isolated boutons or elongated boutons on the type 2 hair cells. When those hair cells are stimulated, when those stereocilia move, then those afferent fibres will send information back to the central nervous system about that movement of endolymph, and then the central nervous system interprets that into defining our position of our head in space and our movement of our head. There is also an efferent nerve supply. These efferent fibres probably alter the sensitivity of the hair cells. They may also amend in some way the information travelling back to the central nervous system by the afferent or via the afferent nerve fibres. The difference between these hair cells in this diagram and the hair cells that I will show you in the organ of Corti, the organ that detects sound, is that there's no kinocilium. And that's because the stereocilia don't bend in any direction towards the kinocilium in the organ of Corti. Their impulse degeneration is due to shearing force and contact with the tectorial membrane that touches them on their apical surface, on the apical surface of these stereocilia. So it's a slightly different method of stimulating these hair cells, but it does not require the kinocilium in the hair cells of the organ of Corti. The crista ampullus, remember, are located, there's 3 of them. They're located in the ampulla or the ballooned or increased sort of bulbous projection of the semicircular canal as it comes in contact with the vestibule. They detect angular acceleration of the head. And what you see in the diagram on the top is the section through the membranous labyrinth within the bony or osseous labyrinth. You can see the bone down the bottom, the osseous component. The crista are projections of the membranous labyrinth into the endolymph of the membranous labyrinth. Remember, there's endolymph in the membranous labyrinth and there's perilymph in the osseous labyrinth. And those projections have hair cells on them, the sensory epithelium. They're called the cristae. The cristae are these projections into the endolymph. And these hair cells have on them a gel-like structure called the cupula. Hard to see in this diagram and also hard to see in the section below that I'll show you in a moment, because getting good sections, good fixed preserved sections of this part of the ear is very difficult because this is embedded in the temporal bone in the head, as you would know. On the lower image, you can see hair cells. These are sitting on the surface of the crista, or the projection into the endolymph. And the cupula is just a very pale yellowish glycoprotein gel-like substance sitting on the surface of the hair cells. And the hair cells, of course, as we saw earlier in the diagram, are supported by supporting cells or sustentacular cells. Very hard to understand really the histological details here, as I said before, because the details aren't preserved that well. Let's go to a diagram to see the details of these structures, the crista. In the diagram, have a look at the projection, the crista into the endolymph, lined by supporting cells and the hair cells. The type 1 hair cells are on the ridge predominantly of the crista. Type 2 cells tend to be towards the centre. You can see nerve fibres coming from the hair cells. Then you can see the cupula, the gelatinous structure that is supported or suspended on top of the hair cells, supported by a glycoprotein called autogelin. That sort of binds the gelatinous structure to the hair cells. And between the two is there's this very fine pores or fluid-filled spaces to allow the hair cells to move. And what happens when there's movement of the head, then there is displacement of the endolymph which bathes these cupula. Remember, they project into the endolymph. And that displacement causes the bending of the hair cells in relation to the kinocilium. They might bend away from the kinocilium, or they might bend towards the kinocilium. And that sends a different message to the brain, as I explained earlier in the lecture. So that stimulation, that movement of endolymph then is what stimulates action potentials from these hair cells and gives information about the movement of that fluid and therefore the movement of the head. The macula are very similar. They have the hair cells, again shown in this diagram. Type 1 and type 2, again they have autogelin, which is a glycoprotein that cements the structure above it to those hair cells. And the structure above it is just like the cupula, except it's called the otolithic membrane. These macula, remember, there's two of them, one in the saccule and one in the utricle. And they detect the linear movement of the head or position of the head. And they do so because the cupula is different here. Or at least I should say the otolithic membrane is similar to the cupula, except that it has within it tiny calcium carbonate structures called the otoliths. So again, when there's movement of the head, these move as well. The endolength moves past these structures, projecting into the ampulla, or the utricles, I should say, and then that moves the hair cells, stimulates the hair cells. The stereocilia again move in relation to the kinocilium, and that again sparks an impulse back to the central nervous system. So the actual process is very similar. The only difference is that the macula have these otolithic membranes containing the otoliths or calcium carbonate deposits, whereas the semicircular canals contain the crista, and the crista are the parts within the ampulla region of the semicircular canal. They have the cupula on them, and the position, or at least the way in which it works, is exactly the same. Here's a magnification, high magnification picture taken through the macula. You can see on the in the image rather, the hair cells, and then they're supporting the otolithic membrane, and embedded in that membrane are these very small calcium carbonate structures, the otoliths, and they move, the membrane moves, and therefore that stimulates the hair cells. So we've looked at the crispa ampullaris, we've looked at the macula that sit in the utricle and the saccule. They're involved with movements of the head, turning of the head, acceleration of the head, etc., position of the head in space because of the movement of that endolymph and the fact that that endolymph then moves either the cupula or the otolithic membrane, and that's detected by the hair cells. Let's now look at the cochlea. And again, it's labelled down the bottom there, number 4. You can see the yellow component, that is the cochlear duct containing the organ of Corti. Here is a section. On the left-hand side, low magnification section through the organ of Corti, and on the right-hand side, a higher magnification. It shows the organ of Corti projecting into the endolymph of the cochlear duct. There's the organ of Corti there. It has a component, or the endolymph is contained within the scala media that you see labelled there. That's the endolymph component of the cochlear duct. There's a spiral ganglion there. The spiral ganglion is the bipolar neurons that send dendritic branches to the hair cells in the organ of Corti, and then the other component of those ganglion cells sends information through the cochlear nerve back into the central nervous system. So that spiral ganglion sits in the bone adjacent to each coil of the cochlear duct. The bone is the medullus that I explained earlier. There is 2 compartments of perilymph, the scala vestibule and the scala tympani. Both those compartments, as I mentioned, contain perilymph. The scala vestibuli starts at the oval window, and the scala tympani starts, or ends rather, at the round window. They contain perilymph, and actually those two chambers come together, and the perilymph passes between one and the other at the very apex of the cochlea, at a place called the helicotrema. And you can see on the left-hand diagram the very apex part of the cochlea. So again, the organ of Corti is embedded in endolymph. Above it, you can see a very small membrane. That's the vestibular membrane. And the organ of Corti actually sits on a basilar membrane, separating the endolymph from the perilymph of the scala tympani. Again, let's go to a diagram for an explanation of how this organ of Corti works. Have a look at the hair cells, outer hair cells and inner hair cells. I'm not going to separate between each of those. The hair cells are strongly, again, supported by different supporting cells, the outer and the inner phalangeal cells. They wrap around the base of the cells and they send a process up to link across near the apex of the hair cells where the stereocilia are. There are other supportive cells there, the pillar cells. They create a tunnel between the pillar cells that support the inner hair cells and the pillar cells that support the outer hair cells. And then the hair cells themselves have both afferent and efferent terminals. The afferent terminals are, again, from the ganglion cells in the spiral ganglion. They're going to take information back to the central nervous system from impulses that these hair cells will send down those afferent nerve fibres. These cells sit on the basilar membrane. And the basilar membrane and the organ of Corti itself is attached to the spiral lamina. It's a thin projection from the malleolus bone. So on the base is the basilar membrane. And the other very important point is that the hair cells themselves are in contact or close apposition to the tectorial membrane above. That tectorial membrane again projects from the bony modiolus. It's a gelatinous type substance. And what is going to happen is that on the base, on the basilar membrane, when that starts to vibrate, it creates a shear force between the tectorial membrane and the hair cells. The tectorial membrane actually touches those stereocilia. And that touching of the stereocilia caused by the vibration of the basilar membrane stimulates a nerve impulse. And that nerve impulse is used to detect sound and frequency and so on. And it's processed by the central nervous system. Well, let's have a look at the real section, a histological section through the organ of Corti. And again, we can label the tectorial membrane almost in contact with the hair cells. Outer hair cells sitting on the basilar membrane there, and the inner hair cells will sit on there as well. And again, as I mentioned before, when that basilar membrane vibrates due to waves of fluid, the perilymph moving along through the scala vestibuli and the scala tympani, created by the oval window being stimulated by the ossicles, this wave of fluid or wave of vibration makes that basilar membrane vibrate also. And as I explain, that vibration then creates a force between the tectorial membrane and the hair cells. And basically what happens is summarised on this diagram. Without going into too much detail, this is a diagram that really unwinds the cochlea. And the white spaces you see there represent the scala vestibule and the scala tympani. The scala vestibuli starts or commences, as I mentioned before, at the oval window in contact with the bony ossicles. The scala tympani ends at the round window. And right at the top of the diagram, there's the helicotrema. That is where the perilymph circulates from the scala vestibuli to the scala tympani. So when the sound waves create the vibration of the tympanic membrane, and that sound energy is then transferred to mechanical energy through the bony ossicles, it causes the oval window to vibrate. And that creates a wave of vibration, or a wave of, vibration through that perilymph. And so the basilar membrane starts to vibrate. And depending on the frequency of the sound, it will vibrate at various locations. It will be displaced. And the maximum displacement component or location will represent the frequency of that sound. And there will be different places for the maximum displacement depending on the different frequencies of sound. And that allows us to discriminate sound frequencies. The basilar membrane detects high frequency sounds down low at the base of the cochlea, and rather low pitch sound or low frequency sounds up at the apex of the cochlea. At the apex of the cochlea, the basilar membrane is its widest, but it's more relaxed or less stiff. So this allows different waves of displacement to be created depending on the frequency of the sound. And the amplitude of the sound will change. The amplitude will create various degrees of displacement. And that amount of displacement will be the way in which we can perceive the actual loudness of the particular sound, not just the frequency. So let me just review the structure and function of the ear. The external ear, particularly the tympanic membrane and the bony ossicles, convert sound waves, sound energy into mechanical energy. And that's transmitted to the oval window. And that oval window then sets up vibrations inside the perilymph of the cochlear duct. And in turn that stimulates the basilar membrane to vibrate, and the hair cells are stimulated as well by their interaction with the tectorial membrane, and therefore we get to perceive sounds. There are also sensoreceptors in the crista ampullaris and the macula utricle and the macula saccules, components of the vestibule and the semicircular canals. And they have special sensoreceptors too, hair cells, having a very specialised function, and the kinocilium. And those hair cells, again, are stimulated by movement of endolymph, either moving the cupula, in the case of the crista ampullaris, in the semicircular canal, or the macula, in the utricle and saccules. And those hair cells, when stimulated, give information to the central nervous system about the position of the head and movement of the head. So thank you very much for listening to this lecture. I hope you now know something about the structures within the ear, their histological structure, and the functions that each of those structures do to bring about hearing and also help us with our balance and position in space.